Hello and welcome to Cellular Biology 101. This will be fun, I guess. In today's class, we are going to discuss the functionality of the eukaryotic cells, specifically organelles and gene expression. Yes, we are going to dive into the mysteries of the cellular core and everything we seem to know about DNA. Firstly, we need to resume the last class, where we understood the basis of the structure of the eukaryotic cell, the semi-permeable membrane, the organelles, and the core. Today we are going to deep dive into them so we can explore the universe of gene expression. Even though every animal species has its own type of cell, we have this cell family called eukaryotic that includes animals and plants. What differentiates us from plants? Easy. Plants have a cellular wall added to the membrane, and we don't. Do you remember what the cellular wall is made of? Yes, cellulose. Quite like starch. Both have a similar chemical structure, but always bad. Herbivores are the only animals that can digest it. Do you remember fiber? Yes, it's cellulose. The other elements are the chloroplast, the organelles that allow plants to transform sunlight, CO2 and water into sugar and O2. Therefore, plants are the base of all life in this planet. As always, I'm rambling. Can't help it. Well, where were we? Oh, yes. Eukaryotic cells. For this class, we're going to treat animal and plant cells as the same type, since we already understand what differences one from the other. Two new concepts we should remember are osmosis and homeostasis. Osmosis is the spontaneous passive process that diffusions of a solvent molecules through a selective semi-permeable membrane. The other, homeostasis, is called the natural process of equilibrium that any living organisms needs to exist, to live. That may include the proper temperature, concentration of micro molecules. Those may be water, minerals, oxygen, etc. Control of pH, etc. In the eukaryotic cells, the membrane shall receive huge amounts of water and sodium, and the potassium may run away. That's why the other magical part of the cellular membranes gets an important role in allowing the cell to survive: the active diffusion. I assume you didn't read this part of the book yet, so I'll introduce you to this concept. Active diffusion is the use of active canals of the membrane that need adenosine 3-phosphate, ATP, the energetic cooling, to work. The reason is quite simple. This canal works against the osmosis principle. Being semi-permeable, the cellular membrane perfectly separates the intracellular fluid, the cytoplasm, from the extracellular liquid. As you may imagine, both intracellular and extracellular fluids must contain different composition of molecules to function. Given this information, what can you infer? No one? Okay. The cell shall produce a lot of energy coins, ATP, to qualify as a living thing. Can anyone imagine what happens when a cell is put in a hypertonic or hypotonic fluid? In the first case, the cell loses more water that it can retain, becoming plasmolyzed and dies. If it's the second case, the cell will receive more water that it can support and explodes. Now, it shouldn't be funny, but try to imagine a balloon. If it has no air, it becomes rugged and flaccid. If it has the proper amount of air, it will do its balloon stuff. But if it has too much air, the balloon will, boom, explode. Moving aside from this image, oxygen enters and carbon dioxide leaves the cell by simple diffusion, osmosis. Like many other macromolecules essential to the cellular function. Now we start to get into the best part of this class. The intracellular space is called cytoplasm. The cytoplasm is made of plasma, water, minerals, proteins, and the cytoskeleton. The last one is pivotal to the shape and structure of any cell regarding the family of them. Inside the cytoplasm resides the organelles. They are connected to the cytoskeleton and use it to move inside the cell. Organize the resources of the cell may need and move the waste it may produce. The principal organelles of any eukaryotic cells are the nucleus, where the DNA resides, the Golgi apparatus, where some proteins are made, ribosomes, where other cellular essential proteins are developed, lysosomes, the recycle organelle, the endoplasmic reticulum, something between the transportation system and a varied drug lab. And last but not least, the mitochondria, the power center of the cell. Today we are going to focus in the nucleus, ribosomes, the Golgi apparatus, and the mitochondria. The latest create ATP from oxygen and sugar. This chemical reaction is called cellular respiration. Random type of the day. Did you know that the mitochondria have the most efficient energetic chemical transfer? It's capable of transform 50% of the sugar energy into ATP. The rest is heat, corporal heat. Since the energetic part of the cells is partially understood, now we will talk about DNA and the cell nucleus. Maybe in the biology class at school, you'll learn that DNA is something like a menu, the most important part of the cells where every recipe resides. Well, even if it's a very simplistic way of understanding DNA, it's true. Every cell in the body has the same DNA, but as the cells are specialized, they only read the fragments that allow them to create the proteins and other molecules they need to properly function. DNA, the desoxyribonucleic acid, is a complex molecule of nucleic acids with a particular structure, the double helix. The said structure is meant to facilitate the replication and the gene expression process. Every nucleic acid has a specific complementary partner in the other strand. Cytosine with guanine and adenine with thymine. Of course, the DNA cannot go out of the nucleus. That's why RNA, the ribonucleic acid, becomes the tool to translate the specific instructions to create whatever is needed. As RNA contains oxygen, It must replace thymine with uracil. Inside the nucleus, the needle extract of the DNA is divided to create an RNA replica that can go to the ribosome and express the given gene. Even though it's a quick process, there are many security protocols the cell will do before letting the RNA getting out of the nucleus. The DNA will be chromatically remodeled. This means the condensed DNA molecules need to be opened for its translation. Once it is, the transcription process begins. The RNA polymerase divide the double-stranded helix and start to produce the primary transcript of the RNA strand as per the complementary law of the nucleic bases. A with U, C with G. The next step will be the mRNA, messenger RNA, and transfer RNA processing. The primary transcripts need to go under a series of modifications to get ready. The most important ones are the enzymatic protection that avoids exonucleases to degrade it, and the elimination of any useless information that may drag out the gene expression process. At the same time, the non-coding RNA maturation takes care of the proper elimination of gene fragments that will meddle protein formation process. RNA export, the final step, once the RNA is ready to leave the nucleus, and the game starts. To me, this is the more exciting part of the gene expression. The principal work of a cell is to produce proteins. Yes, specialized cells will produce other molecules, but the base is the same. Please remember, mRNA eukaryotic cells will only carry one protein instruction. Continuing with the travel of the mRNA and the tRNA into the protein assembly. It's the translation. In this part, the mRNA will travel to the ribosome to be read, and each tRNA will carry one amino acid particle into the assemble inside the cell organelle, creating a chain of amino acids. The tRNA is formed of three nucleic acids that will match perfectly with the large strand of mRNA. This is how the first step of the protein is done. Sadly, the protein at this point is useless. Once the chain is perfectly created, it needs to take a 3D structure to function. This is why the folding and translocation comes to the rescue. The chain of amino acid goes into the Golgi apparatus to get folded. This means the hydrophobic parts of the protein start to agglomerate, and the hydrophilic part of this will protect them. Once the process is done, the protein will be translocated to the secretory pathway so they can be used. The final step of the gene expression is a protein transport. The proteins are ready to do their job in the organism. It may vary from getting out the body in from or hair, affect another part of the body into enforce something, attack an intrude, etc. There are great questions we should answer. How is the making protein process related to gene expression? How do the proteins determine our height, skin tone, hair, eye color, etc.? Our bodies are made of proteins, from the cells to the external structures. Even if the cell doesn't produce a specific proteins, like chloride acid in the stomach, the process is the same. And it may be the cell needs a protein to produce another element, some like hormones. Many of our characteristics in our bodies are dictated by proteins our cells make. The color of hair is dictated by pigments added to the keratin, a protein. The color of our skin depends on the quantity of melanin our body produces, another protein. Proteins are the basis of our existence, and that's why we should remember this process.